The aim of this study was to develop a working protocol to reveal the neural activation pattern underlying the flag task by using fused EEG and fNIRS neuroimaging technique. The simultaneous fNIRS EEG recordings allow for the inspection of the relationship between the hemodynamic response of prefrontal cortex and different event-related potential components of the whole brain. fNIRS measure the acid and deacid hemoglobin concentration changes in the cerebral cortex, which play an important role in study of cognitive processing. We know that fNIRS is mostly used for inspecting the neurovascular activity in the frontal lobe that is related to high brain cognitive functions. However, the hemodynamic response measured by fNIRS can only indirectly capture the neural activities with lower temporal resolution, but EEG can offer temporally fine and direct measurement of neural activities. The combination of EEG and fNIRS recordings is able to identify more features and reveal more information related with the brain functions. In this study, used EEG fNIRS techniques were used for simultaneous recordings of ERP components and hemodynamic response with a flag task. It's rational to assume that